So let's get into the agenda for today. So it's Halloween. It's a bit of a weird Halloween because we are mostly inside. Um, I hope you guys are, if you're going out and making plans, I hope you're practicing like social distancing and stuff like that. Uh, but some people have plans, some people don't. Whatever it is, we'll talk about it in the opening segment. We'll share out, see who's doing what, and then we can move on into the main activity, which will be the outline guides. And some of you may be familiar with this because I did it in my first alternative virtual SI session, where I essentially looked at all the material you guys covered in class, and I broke it down into the parts that you really need to know so that you don't have to sift through all of the boring or more specific nuanced information that you won't need on the test. And then our final activity will be a resource referral activity. So essentially, I'm going to have an open slide, and everyone in here will be expected to contribute uh, either one or maybe multiple resources that you are used to using for these exams when you're studying, and how well they work for you so that other students also have the chance to utilize the resources that they don't know about. And so I find we have a group of students like across the board together, there's a lot of good resources that you guys use, but sometimes you don't know what all those resources are out there, and so your fellow students can help you with that. With that being said, why don't we get into the bulk of today's review? So, first things first, we have animal history and development. So this is a bit of a combination of the first, second, and third lecture broken down into different bits and pieces that are really important. And so I kind of split it up into animal history, or animal evolutionary history is what it really is, and animal development, because those are two big umbrella topics, I feel, that will help you guys separate information and also understand how the information connects within those like smaller bubbles. So why don't we start with animal history? So the first thing is defining traits of an animal, right? We know that when we're talking about life, animals are distinct from a whole class of other animals like plants and bacteria, because there's specific things about them that differentiate them. And so the things we usually talk about are they're multicellular, right? They're eukaryotic. And so that separates them from like bacteria, which might be prokaryotic. And then you also have their heterotrophs. So they don't, produce their own energy by making their own food or anything like that, unlike plants, which do, autotrophs that do that. So that's separate. They also are able to move, right? They move around to mate or to get food. They have some movement, and that doesn't necessarily mean that they move from the spot they're in, but they have moving parts within them that help them obtain food that they need. Also, they have reproduction that's a little different from, other types of organisms, but that doesn't necessarily mean that they're only sexual reproducers. They can be asexual, or they can be hermaphroditic. And but a really convenient thing that we know is that animals are all in a monophyletic phylogeny when we're talking about the grand scheme of organisms and life on Earth. So they're not like split up in different branches or anything. They're in a smaller subsection of eukarya. They're all on their own. And so it's pretty easy to keep track of animal development through history, since they're all under that one monophyletic group. And so when we're talking about animals and how animals, you know, essentially become more diverse over time, we have to think about different factors of diversity, and we have to think about different types of evolution. And so the first thing I have here is divergent and convergent evolution. You guys should already know this from previous units, especially Unit 1, where we're kind of talking about genetics and stuff like that. And even Unit 2, because it still works with plants. But essentially, um, based on similar factors in similar environments, certain animals can evolve and develop similar traits or characteristics that make them look alike and have similar functions, but doesn't necessarily make them related. It just means that they have similar solutions to problems that their environments push against them. Or divergent evolution, animals in different environments obviously need to form different uh, functions so that they can survive as best as possible in their own environments. So those are the two main types of evolution. But we also have to think about what drives those evolution, two types of evolutions. So you've got diversity factors that help distinguish between animals, including one, so feeding types we talked about. There's different feeding types among animals. You have suspension feeders who, just suck in a lot of food and then filter out whatever they don't want. And so an example of this is like a baleen whale. You have deposit feeders who, kind of rummage through surface areas and mediums, essentially to get nutrients from like dirt or soil. And so you can think of like worms as deposit feeders, fluid feeders. So these are more like insects that use the nectar of like flowers and stuff like that, or even like a hummingbird, which feeds on the fluid of flowers and the nectar of them. And then you've got mass feeders like us humans, but mass feeders is a little more complex because then you also have, herbivores who kind of mash food because they're mashing, they need to mash plants that they're eating, or you have carnivores who have sharper teeth and they need to rip apart muscle and fat that they eat, and omnivores like us because we have the mashing and the ripping ability because of the way our teeth are formed. Then we also have the diversity factor of sensory mechanisms. There's a whole lot of them. Uh, obviously you have the five senses: touch, smell, sight, hearing. So all of that kind of stuff. Um, but then you also have other stuff that some organisms have, like magnetism. Turtles know where the poles are and can navigate their way in the ocean based on that. Or, electric fields. Sharks can sense electro impulses. You have birds that can detect barometric pressure. And you have some animals, like some snakes, can read thermal temperatures. And so judge like targets or environments off of that. And so then we go into locomotion finally, which is the distinction of how animals are mobile. There's lots of different forms, obviously, right? Animals fly, animals swim, and then even among like different categories of like land animals, you have jointed limbs, you have tubes, you have bristles. Um, so there's all sorts of stuff there, and that kind of plays into the diversity of the different environments that are available in these different mediums. So just something to keep in mind. But when we were talking about early animal evolutionary history, what we really talked about was the like more early and distinguishable life forms that we can focus on. So sponges, one of the earliest ancestors of the animal phylogeny, one of the earlier organisms present in it, can be distinguished from later organisms because it doesn't have any form of symmetry, and it's also monoblastic, so it only has the endoderm, which is the like internal uh, tissue for them. Then you have stenophores, which branch out from sponges. They have radial symmetry, and they're diploblastic. So they have two tissues. They have the endoderm and the ectoderm, internal tissue and external tissue. Then you have the Parahoxoa, which is more closely related to us, but we're a little far down from there, a little farther down the line in Parahoxoa. Essentially, you have a development of bilateral symmetry, and you've got tripoblastic defining traits. So essentially, not only do they have an endoderm, which is internal tissue, and ectoderm, which is external tissue, but you also have a mesoderm, which is in between those two in the middle. And at Parahoxoa, you also have a differentiation that splits down into protostomes and deuterostomes. Protostomes, if you guys remember, in early development, their blastospores develop into the mouth, whereas in deuterostomes, for early development, the blastospore develops into an anus. So the distinction between those groups is very important to remember. And so I was talking about the three different tissue layers, but it's probably important to know what those different tissue layers are responsible for as well. So the endoderm is the very internal tissue layer, and it is responsible for digestion and also respiration. And so obviously this is very important for early animals to have because they need digestive capabilities to get food and to get energy from that food. Then we do have the mesoderm, so only present in tripoblastic creatures, which is responsible for the muscular system, circular system, and skeletal system. So it's really about like the infrastructure and support within the organism. And this is not as important um, as the other two because uh, it depends on what environment or medium you're operating in, whether or not you really need those kinds of additions, right? If you're in like the ocean and you are like a somewhat um, less mobile organism, then you really don't need as much in terms of a mesoderm. And then finally, we have the ectoderm, which is essentially the external layer or the outer layer on organisms. Think of it like your skin. And it also is responsible for the nervous system, which is very important because it has the ability to trigger responses and also respond to stimuli. So that's an important development as well when we're talking about that. And then we get to a little more specific stuff within the whole Parahoxoa um, tree of the phylogeny, where you have some animals have hox and parahox genes, or it essentially comes from the word homeobox. And what a homeobox is essentially a set of genes or sequences that are fundamental to determining the development of an organism in its embryonic stages. And so the homeobox can be really useful in determining like how the adult form will develop, like what functions and what limbs might develop, as well as comparing between species, the similarity in homeoboxes, because that can help us determine similarity between organisms. And then you have the notochord, which is also a development, and it's essentially a cartilaginous skeletal rod of support for the organism. And it's basically the precursor to what you would consider as the spine. But the important thing to note is that even if an organism has a notochord, that does not necessarily mean that it will develop a spine or that it will become a vertebrate. essentially have a bony spine or bony structure. Because when we're talking about the phylum Chordata, which the notochord is specific to for a lot of creatures, the chordata only specifies that creatures have the notochord at some point in their lifetime. So that doesn't mean that they have to have it for their whole lifetime. So just because a creature has a notochord does not mean that it is going to be a vertebrate. There are some notochord organisms that are invertebrates. And so at the big picture of the tree that we looked at, we kind of have, sponges versus stenophores, because that's the first big gap and change where there's a development of symmetry and there's also an addition of a tissue layer. And then there's also cnidarians versus bilateria. So cnidarians, they're the first group that get the Hox, parahox genes. And so that's a big addition there. And then bilateria, they add on a sort of bilateral symmetry, which is much more specific than the radial symmetry that you see in cnidarians. And so bilateria develop a little more from there. And bilateria are furthest down the tree that we looked at when we're talking about early evolutionary history. Cool. All right. So that was the first part. That was animal history. I hope you guys are hanging in there. I know it's a lot to cover, but hopefully like having this information condensed in a neat outline will help you understand kind of how to navigate the material and how to look at it in a more speedy way. So now we can move on to animal development, which is the next, big thing in terms of how we look at animals and how we differentiate between animals. Now that we're not looking between, animals and other organisms, we're just looking in the class of animals. And so there's 3 different things that you can look at the beginning stages of an animal in embryonic development. There's cleavage, which is essentially like the division of cells and separation and multiplication. And, that is very dependent on what organism we're talking about. So it's specific to the organism and just important to know what it is, but not really important to know like what groups of organisms have cleavage where and when, because it's pretty organism specific, species specific. Then you have gastrulation, which is essentially what we were talking about earlier, how protozomes develop a mouth from the blastospore and deuterostomes develop an anus from the blastospore. So gastrulation is kind of the stage where that determination happens in an organism. And so that's kind of the separation point between protostomes and deuterostomes. And then finally, you have organogenesis, which is basically the creation and development of the organ systems in the organism. It doesn't matter where, it's basically the entire network. And so obviously animal organs can differ a lot between species. And so this is another important step for determining differences between animals. Yeah. So going back to what we were talking about, we have animals need to manage the different like levels that they have within themselves, and they also need to manage the materials that are in them, as well as be able to obtain some nutrients or some chemicals and molecules from outside. And so they have a diffusion, and depending on the environment that you have, you're going to have a lot more diffusion in an animal or a lot less. So obviously in like an aquatic environment, you'll probably have more diffusion slash gas exchange between the environment and you, because it's like a lot more open and fluid. But an important thing to remember is diffusion is not osmosis. They are 2 separate things. So diffusion is all about like different, nutrients and chemicals and the gas exchange between the animal and their environment. So it's an important distinction to make. Then we get into the hierarchy of the animal system. So by now, I'm sure a lot of you guys know what this is or what the order is, but essentially, most complex, you have the animal, then you have the organ system, then you have the organ, the tissue that makes up the organ, the cells that make up the tissue, and then the organelles in the cell, and then the molecules in the organelles. And it's not too hard to remember just because it's pretty easy to visualize how your body breaks down or how animal bodies break down, but it is an important thing to remember. And there might even be a question on it in the test. I wouldn't put it past Dr. Wilkin to put a question about that. So, you have all of these complex levels in an animal, and you know, all of this is really great, but there has to be some sort of maintenance for all of this complexity, right? There needs to be some sort of system that controls the environment that's inside the body. And so here we stumble across the next part that we want to talk about, which is homeostasis. And homeostasis is essentially the body regulating an internal environment between some extremes or within a control to make sure that the optimal levels of chemicals or pH or whatever, there's a lot of things that homeostasis covers, is maintained to optimize like enzyme interactions and functions and processes so that they continue working. And so when we're looking at homeostasis, we look at a couple things. We look at, first of all, bioenergetics, we talked about, which is essentially the study of how cells transform like the food that they get into energy. And so that's a really important part of homeostasis because you need to maintain this sort of energy in your body to continue functions with your muscles and movement in your systems, breathing, etc. And then we talk about thermoregulation as well, which is also highly related to bioenergetics, which we dove a little more deeper into because it's got quite a few facets. So essentially what we do is when we're talking about organisms, we can usually break them down into two groups. They're either endotherms or ectotherms. And so endotherms are creatures that essentially uh, have an active metabolic rate and metabolism that they use to maintain the temperature and also like system functionality within themselves. And so they do not rely on their environment for the temperature and heat that they need. They make sure to do that on their own. And so that's what endotherms are. Whereas ectotherms actually rely on their environment to bring them the heat that they need. And this is kind of what we talk about when we're talking about warm-blooded animals and cold-blooded animals, right? Like most reptiles usually fall under the ectotherm. Even fish as well are mostly ectotherms. And we have like mammals and birds, which are mostly endotherms. And the good thing about endotherms is because they maintain their own metabolism and don't rely on their environment for heat, they can operate as they wish, and they don't have to be concerned about uh, trying to find heat or being in unfavorable environmental circumstances. But the problem is keeping an active metabolism requires a lot of energy. And so that's why ectotherms have the benefit of they don't keep up an active metabolism. And so therefore, they do not spend as much energy on keeping their body temperature consistent as much as possible. But the problem is because they rely on their environment so much, their behavior is shaped by their needs. So essentially, if you need to get heat and you're an ectotherm, then you do have to spend like a lot of time in the sun to make sure that you are maintaining your internal systems as best as the environment can allow you to do so. And then even within endotherms and ectotherms, we have a little breakdown of heterotherms and homeotherms. So heterotherms, they allow their bodies to fluctuate between temperatures. And so most ectotherms or most heterotherms are exclusively ectotherms, just because ectotherms rely on their environment to manage temperature. So they're not really keeping their body consistent always. It all depends on their environment. And then you have homeotherms, usually correlate with endotherms, but there are some ectotherms that are closer to homeotherms. Essentially, homeotherms are the opposite of heterotherms because they maintain their internal temperature and body as close to constant as possible. And so Dr. Wilkin had a great chart about that in her slides. So I would highly recommend looking back at that if you want some insight about that a little more, because that it does a really good job of illustrating kind of how these species separate based on thermoregulation. And then when we're talking about thermoregulation and metabolism, we also come across this conundrum that sometimes people get confused about, where smaller organisms spend more energy per gram of tissue because they actually have fewer cells, and so each cell is doing a lot more work, and therefore requires a lot more energy to maintain. But that doesn't mean that they will spend more total energy than a larger organism. The larger organism will always spend more total energy, but smaller organisms always spend more energy per gram of tissue because each gram of tissue and each cell requires a lot more maintenance. So remember to keep that in your minds so you don't get confused about it on the test. Always look for that key phrase, per gram of tissue, to make sure that you know what the question is talking about. And then we talk about differences between like aquatic creatures and also terrestrial creatures. And terrestrial creatures also include like aerial, like birds and stuff like that. But so in these different environments, there's obviously different challenges that are posed by the circumstances. In an aquatic environment, there's a lot less oxygen abundant and available to like fish than say a human on land, because oxygen is pretty easy to secure. It's already out there as an O2 molecule, but oxygen is bound to hydrogen atoms in water as the water molecules, so it's a lot harder to come by. And so the sort of solution that aquatic organisms have and fish have specifically is that they have like a counter current flow in their bodies, where essentially they keep their mouths open and water flows into the mouth and then out of the gills. And during that process, oxygen is removed from the water and is directed into the bloodstream. And so by keeping that countercurrent flow, the fish are able to cycle out water and obtain oxygen, without like becoming oxygen deficient in any way. And they are one of the, it's more common to find a countercurrent flow in an aquatic creature than it is a terrestrial creature, because obviously you don't have a problem like that in the terrestrial setting. So, but yeah, water flows in the opposite direction that the blood with oxygen does in the lamae. And that helps oxygen uptake into the blood because of the concentration gradient, and some diffusion as well. And then another important thing to think about fish and aquatic creatures is they tend to have a unidirectional flow in their system. So deoxygenated blood and oxygenated blood flow in the same direction. They're only in one circuit. Whereas opposed to all other creatures, all terrestrial creatures at least have two circuits and generally tend to only have two circuits. So that oxygenated blood can flow one direction and deoxygenated blood can flow another. And so when we're talking about how there's double circulation in terrestrial creatures, we talk about, first of all, the respiratory system. A lot of the oxygen is captured in the alveoli, especially in like creatures like birds, which have even more air sacs to optimize the amount of oxygen that they capture, especially at high altitudes where oxygen is thinner. And then you also have the fact that the circulatory system manages the blood in a very clean and organized way. So essentially, you have the pulmonary circulatory system and then the systemic circulatory system, where deoxygenated blood will enter the heart in the right atrium from the body, and then it will go to the right ventricle, and then it will go to the lungs, where the lungs will release out, like carbon dioxide, send it back into the air when we exhale and stuff like that. And then on the systemic circulatory path, you have oxygenated blood, which comes into the left atrium from the lungs. So reverse of the pulmonary path, right? Pulmonary path, it's from body to lungs. Now it's lungs to body, where essentially the blood comes from the lungs to the left atrium, then it goes to the left ventricle, and then it flows out to the body. It's distributed that way so that you get the oxygenated blood to all the parts and muscles that you need. And so that's the kind of breakdown of the circulatory system. I put pulmonary right, systemic left to remember how, like, which side deoxygenated blood is on and which side oxygenated blood is on, because they operate on two different sides, and it's pretty easy to separate them that way. All right, so now we're on to the last two lectures that you guys covered. These ones are a little more simple, and they are, a lot easier to wrap your head around, just because the information is uh, a little less condensed and a little, simplified in a sense. So the first thing we want to talk about is animal osmoregulation and excretion. Essentially, how do animals balance the water levels that they have within themselves with the environment that they're interacting with, and as well as how do they get rid of waste. And so an important thing to remember from the early examples that we talked about in class is a freshwater fish is hyperosmotic to its environment, and hyperosmotic means that it has more solutes per water, so more concentrated water, more concentration in the water than its environment. And so a freshwater fish has more solutes in its body than the freshwater environment that it lives in. And contrastingly, a saltwater fish is hypoosmotic to its environment, so it has a lesser concentration of solutes in the water in itself compared to its environment. So those are the key distinctions between these two types of fish and how they interact with their environment. And then you also have an isoosmotic situation where the concentration of solutes are the same within two mediums of water that are interacting with each other. And that's generally the goal of osmosis, to essentially balance out the concentration gradient. And so when we talk about osmoregulation, similar to thermoregulation, you have two types of organisms. Organisms who are osmoregulators, who have an active function and process that helps them maintain the water levels that they need in their bodies and helps them not lose water or nutrients to their environment. And then you have osmoconformers, who kind of like passively interact with their environment and let the environment help them with nutrients, or remove nutrients from them. They don't really have control over it. They just need to be in a place that has a good amount of either water or solute depending on their needs. And so that kind of correlates to waste in animals. Essentially, if you're able to control the amount of water that you have, or at least you're in a good area with plenty of water, it can really help with what kind of waste you produce. So ammonia is a waste that's typically produced by like fish because there's a high water requirement for the dispersal of the ammonia and release of it. And it's also highly toxic compared to the other waste products. So it's pretty specific to aquatic creatures, but there are some terrestrial creatures that produce ammonia. They just usually also have like high interaction with moist environments and stuff like that. Then we also have urea. So there's less water needed, and it's also lower in toxicity than ammonia, but it requires much more energy to produce, just because you don't have as much water being able to direct the flow of concentrated waste. And so humans, among many other animals, such even like sharks, tend to actually produce urea as opposed to ammonia or uric acid. And then finally, you have uric acid, which requires very little water, has low toxicity, and requires more energy as well. And this is typically waste released in sort of like egg forms. Essentially, if you're an animal that's in like a low water climate, not even close to water, um, then you'll probably produce uric acid. So like a lot of desert animals produce uric acid, and also some reptiles. So that's an important thing to remember. Finally, there's another important part with how animals regulate the water that they have within themselves. So the kidney is the organ that's most responsible for the maintenance of water levels within the body, as well as nutrient levels within the body. And so a lot of the ways that animals are able to conserve water or lose water correlate directly with how the kidney performs. And an important thing to remember is a loop of Henle. A loop of Henle is part of the process that kidneys dictate. And essentially, it's the part where the body can communicate with itself to either create more concentrated urine or more dilute urine based on the water levels that are already existing within the animal and what is needed of the animal. If you have a longer loop of Henle, there's going to be more conservation of water because there's also more surface area for the water to be retained and reabsorbed during waste removal. And there's also the important chemical ADH, where ADH can be directed by the nervous system to essentially um, either bind or be held back. If ADH binds, it makes the water duct highly permeable. And essentially, what that does is that helps with a lot of water retention when waste is being removed. And so, if you need to conserve as much water as possible, as an organism, then ADH is also a useful chemical for doing that when you are removing your waste as well. And so, yeah, that's pretty much osmoregulation and excretion. Pretty simple stuff. I think probably the easiest part of the unit that you guys covered, probably the easiest to understand. Then you have animal locomotion and anatomy. So it's also a short section, but it's also got a little more complexity to it. The important thing being that locomotion is obviously how we understand movement in animals. And it's very important because animals need to find mates. They need to, catch prey or find food resources that they need, depending on if they're like a herbivore or a carnivore or an omnivore. And then they also need it to travel around like to different habitats and stuff like that. So yeah, it's very important for animals to have the ability to move, and depending on their ability to move, like what kind of terrain or environments that they can traverse. And so we kind of break it down into two big systems. You have the skeletal system first, basically all of your bones and stuff like that. And the skeletal system performs a very important function. Um, First of all, it's the main structural support of the body. So with the skeletal system, you're able to like shape muscles around it and essentially develop the template for where muscles need to be laid out so that they can perform the various functions that help with movement. Then they also, protect the vital organs that you have. So your skull protects your brain, obviously. Your spine protects your spine and your ribs kind of help protect your like lungs and stuff like that, and all the internals that you have here. So the skeletal system is really important in that regard. And although it's not like as important in movement, it's important for laying the foundation for movement in animals. And then we have the muscular system, which is basically all the muscles that you have that help you contract, whether it be your biceps, your triceps, your quads, whatever it is. They're all over your body and they surround your bones. And the important thing to remember about the muscular system is essentially how muscle contraction happens. And so when we're talking about muscle contraction, essentially what you have is there's always going to be two muscles that act as a pair for any contraction. If one muscle flexes, then the other one has to relax. You can't have two muscles, flexing at the same time, because then you'll just have a rigid or restricted um, limb. But having one relaxed and one flexing allows for movement in a certain way. And so the way that muscle actually moves is broken down by the sliding filament theory. The sliding filament theory essentially proposes that you have these myofibril filament structures out here, essentially thin muscle fibers that are lined up along like this. And then you have the myofibril also has like tropomyosin in it, and it also has actin in it, and calcium actin binding sites on it. So they're like kind of the outside piece of the contraction. And then you have the inner piece, which is a big like strand of a sarcomere with myosin and myosin heads. They're essentially like bulbs at the end of like strings. And what happens is, so ATP helps to hydrolyze myosin heads, which essentially means it helps it lift off from the sarcomere and extend towards the myofibril structure out here. And then what happens is, because the myosin heads are out here, they're able to bind and cross bridges over to the actin or the actin binding sites, which are on the myofibril. So that's really where the myosin and the myofibril connect at the actin binding sites, where the myosin heads bind to the actin. And then because of those bridges, the myosin actually rotates the sarcomere, essentially bring it forward or backward, depending on what movement that you need. And that's really the movement that your muscle experiences. And then what happens is the myosin again binds to ATP, and that helps it separate from the actin binding site. So that detaches it from the actin. And that's essentially the whole cycle in like 4 steps, right? So there, I have it down here. There's actually these 4 steps. You have rest, where nothing's happening; excitation, where the ATP allows the myosin head to reach up to the actin binding sites and grab them. Then you have contraction, where the myosin heads rotate the sarcomere to move in a certain direction. And then you have relaxation, where the ATP attaches to the myosin again to break it off from the actin and to release the hold that it has. And so a couple of these movements helps your, filaments slide along each other. And so the thick filament is really the sarcomere and the myosin inside, and the thin filament is the myofibril. And that's why it's called the sliding filament theory, because it looks like the thick filament slides along the thin filament as the myosin heads move along the structures. And so an important thing to remember in this situation is that the sarcomere is the only part that actually shortens for contraction, but filaments always remain the same length. And then calcium ions that are floating around can also block contraction because they can actually lock onto the binding sites of the actin instead of allowing myosin heads to bind there. So that's an important way that that can be stopped. And so the final thing about locomotion is that obviously there's a cost of transport um, that we talk about when you need to uh, contract your muscles and move around, because every movement, uh, simple or big, requires that muscle contraction that we talked about in the sliding filament theory, and that obviously requires ATP at multiple steps for it to continue in a cycle. And so uh, this transport can be very costly in energy, but that energy cost also depends on the method or the medium that transport is being taken in, as well as the size of the creature. And so this is another chart that you should go and look back at in Dr. Wilkins' slides at the end of her last lecture. Essentially shows that as size increases, whether you're swimming, whether you're running, or whether you're flying, you will have a smaller transport cost than you would have if you were a bigger size, and that's just because, again, the smaller the smaller organisms have more work to do in all of their cells because there are less cells, and therefore they have higher transport costs. And so that's a big takeaway about this that kind of relates to the energy loss that we were talking about earlier between small and big organisms. And at a certain point, it also intersects, the transport costs also intersect, or they're also parallel in their slope. So those are all things to keep track of when you're talking about that. Cool, yeah, so that's all we have to cover for review.